I'll try to be brief. So I want to talk about the wastewater surveillance project I did. This project encompassed metagenomics, which is the study of entire communities of microbes within a sample. Okay, so the goal of the project was to identify the sample composition of wastewater samples that were collected by the members of the lab. And I wanted to follow the pipeline suggested and developed by Nanopore on their website. So pretty much what I was doing, I got the raw hot5 files that were generated from a single flow cell. I did base calling, I did some quality filtering, and then on this quality filter reads, I did direct taxonomy classification. I also attempted to do de novo assembly of the reads, after which I wanted to also do taxonomic profiling of the assembly that I just assembled together. So sample details, I had 3 samples total. Each of them was about 100 gigabytes, and they were collected from wastewater, as I said before, I think in New York City. And obviously they were sequenced using Oxford Nanopore technology. First, I did base calling using the DNA base calling model. I used Dorado, just like we did in class. And just one note here: the Dorado DNA models do not support modification calling, so I had to do it on the canonical base caller without any methylation or other modifications. But I still had a lot of reads to work with. So first, I did some quality filtering. I excluded very short reads, less than 200 base pairs, and I also excluded reads with pretty low threat, PHRED score of lower than 8, which means that on average 1 in 8 base calls would be incorrect, which at the beginning I was trying to be a little bit more conservative. I was trying to keep a little bit longer reads and have this higher threshold for threat score, but then I was not getting this many reads. So I loosened my restrictions a little bit. And also what I tried to do, because we are analyzing wastewater and we try to detect different microbes, and I was not interested in human samples. So I aligned all of my reads to human genome, HG38, and removed all the reads that aligned to human genome. At the end, out of the 3 samples, I was left with over 100,000 reads for each of them. So I could proceed to some taxonomic classification. I used a tool called Kraken, and even though I attempted to exclude the human reads, 95% of them got classified as Homo sapiens using Kraken. So I was very disappointed and didn't really know what to do with that. But I also identified a significant presence of the bacteria called Streptomyces, the bacteria of genus Streptomyces, which is ubiquitous in soil and aquatic sediments. So I was not surprised to find them in wastewater. However, again, most of my reads were identified as Homo sapiens. And I also tried using BLAST to also do some taxonomic classification using a different tool. But again, most of my reads were getting aligned to Homo sapiens. So I was very disappointed, because even though I tried to exclude human reads during quality filtering step, I was still getting so many of human reads. So I thought, maybe let's use these human genes. I wanted to identify the most abundant genes of humans that I identified in my sample. I did not run the whole gene expression analysis, because that was not my goal. I just briefly looked at them. So the x-axis is not really meaningful. It's not normalized, but I just looked at absolute scale, absolute raw count of all of my genes. And I found this results pretty interesting. Like, you can use this kind of gene detection to, for example, like use human tissue specific markers to point to epithelial cell shedding, or just reflect human biology at the population scale, or also it can serve as population level disease biomarker. So in this particular example, for example, link 00486 is highly expressed, and in general, this gene is highly expressed in colon or bladder, and its presence in wastewater may reflect gut or urinary tract epithelial turnover, which I found pretty interesting. And then I was like, you know what, screw the filtering. I would just run all of the reads I got from the base color through the taxonomic classification model, and then I identified a little bit more species of bacteria. Still, most of my reads were classified as Homo sapiens or were unable to be classified, but now I got a little bit more species of bacteria. And these results I also found pretty interesting. A lot of these bacteria are pretty common in different environments and can survive in very diverse habitats. And this, I couldn't really get anything meaningful out of it. I just kind of saw what the species are, but I think more deep studies can have very, very good, maybe a good idea to just monitor pathogens, because some of these bacteria may carry some false implications for humans. We may also study some of these bacteria to study their role in enhancing organic matter breakdown, or a lot of different, there can be a lot of different uses for this kind of analysis. After I did all of that, I wanted to take all the reads that I passed through this quality filtering and that I identified, and did some de novo metagenomic assembly. I did it using the Meta MBBG tool that was suggested by Nanopore, and this ran for I think 10 hours. So, and it finished this morning. So I was very nervous that I wouldn't have any results, but thankfully I did. The tool managed to identify 59 contigs, and contig refers to a continuous stretch of DNA sequence that has been assembled from overlapping DNA fragments from all of our reads. They were pretty long, however, no contigs over 1 megabase, which from I read is pretty typical for wastewater samples, for metagenomic samples. And as you can see on the histogram of contig lengths, most of them were relatively long. So I assumed that I would get something interesting out of the taxonomic classifications of my de novo metagenome. However, after I ran it through the taxonomic classification tools, they all classified all of my contigs as Homo sapiens. So that was a bummer, but I did this morning, so I couldn't really run it again to see what was going on there. So I was very excited for these results, and this morning I was very disappointed. And also my de novo assembled genome failed. The tool that I tried to use as its quality check that was supposed to tell me its completeness and contamination. So I think this project requires deeper digging and maybe working on it a little bit more, but I found it really interesting to work on this type of things. And I think my main takeaway is that human DNA is abundant in wastewater samples. There is a lot of human biological material in wastewater, like skin cells, hair, bodily fluids. And it usually, from what I read, overwhelms microbial signals. And wastewater surveillance is very important because it has potential for population level health surveillance using human gene markers, but also if we exclude human samples, maybe more successfully than I did, we can look at bacteria and microbes that are there and maybe make some conclusions based on that. So thank you very much.